Hello. So this is gonna be to talk about the third genetics exam. Oh my gosh, give me a second. Okay, the third genetics exam is gonna be about the central belief or the dogma of genetics, and that is basically DNA going from chromatin to RNA and then to DNA. So basically, DNA, RNA, protein. Wait, I think I said something wrong. Well, basically, it's basically DNA, RNA, protein. That's a really simple way of putting it, but that's basically the simple central dogma of genetics. So we're just going to look into it in a more like in a deep way, and in the end, we'll look into the revised central dogma that I kind of put in my own words. Okay, so starting off, all humans basically have two autosome pairs. If you remember from last lecture, it was basically 22 autosome pairs are chromosomes that do not have anything to do with your sex. And then you have one allosome pair. So 22 plus 1 pairs, that means 23 pairs. Then you have 46 chromosomes from those 23 pairs because they're pairs. So 23 times 2 is 46. And with those 46 chromosomes, it will result in at least 6 million base pairs for all of your genome. So that's a pretty crazy amount of like genes and base pairs that can code for amino acids. So the main like packaging of DNA is in chromatin. And this is basically condensed DNA and proteins or histones during metaphase. So this is like the generic packaging of DNA. You have two types of chromatin. You have euchromatin, which is tightly packed. Lightly packed. Euchromatin is lightly packed and rich in gene concentration. And since it is so lightly packed and open, this part of the chromatin can be used for gene transcription. Now, heterochromatin is tightly packed and is genetically inactive, so you can't use that for transcription. So there's two types of heterochromatin. You have constitutive, so this is the type of heterochromatin you'll find at telomeres and centromeres. These are very, very gene poor. And then you have facilitative heterochromatin, which are basically like involved in gene silencing. For example, bar bodies. And bar bodies are mainly in females, in women, because you know how women have two X chromosomes, so it's XX. Only one of those chromosomes is expressed. So, for example, for males, you have XY. The important chromosome in males is the Y chromosome. Now, in females, you have an important X chromosome, but you're not going to use both of them. So, one of them is going to be inactive. That's the bar body. So that's a facilitative heterochromatin. And then constitutive heterochromatin is just in all chromosomes, you know? Anyways, you have a couple of DNA and protein association models. So these models basically show the construction of protein from DNA. So there's two types of models. The first model is the folded fiber model. This describes chromatin as like coiled and folded fibers, and it compacts DNA into a 300 nanometer fiber with each chromatid consisting of a single fiber. So this is probably common in seeing some like cell cycle diagrams and whatnot. And it's constructed of type A fibers, which is 1 to 10 nanometers, and then type B fibers, which is 20 to 25 nanometers. Now, the second association model for DNA and protein is the nucleosome model. This is the most and most widely accepted model of the chromatin, and this describes the nucleosome as the molecular unit of chromatin. So basically, what this is you have proteins called histones, and think of these as like Tums or like Tic Tacs or Smarties, right? And then you have DNA that's wrapped around those histones. So think about Twizzlers or like sour strings, and those are wrapped around like the Smarties. So it looks like beads on a string, basically. So these is basically that's a nucleosome. And the nucleosome does have localized areas for transcription and protein biosynthesis. So that's pretty interesting. So let's just say we're going to go with a nucleosome model. There's a couple types of histones. So you either have a core histone or a linker histone. So first of all, we're going to talk about core histones. This includes H2A, H2B, H3, and H4. You're like, where's H1? We'll talk about that in a bit. So core histones are about 120 amino acids long, and these are very, very highly conserved during evolution. And these forms, sorry, they form follicles. Sorry, I can't pronounce. And these are very basic. So it's 25 lysine and arginine. And then H1 is a linker histone. And these are about 200 amino acids long, and they're not highly conserved. They're tissue specific, and they're loosely associated to the core DNA. So basically, the difference between core and linker histones is like core is actually involved in the DNA sequence, like the chromatin sequence, and linkers are only like they only help connect or like connect sequences like nucleosomes, or they help with higher order coiling. So that's basically the difference. Okay, so now we're going to talk about how euchromatin is turned into heterochromatin. We're going to talk about 3 types of processes or processes in this lecture. So basically, first we're going to talk about nucleosome formation, or like how euchromatin is converted into heterochromatin. So basically how a loose chromatin is converted into a more dense, highly structured, and more complex coiled chromatin. The second part we're going to talk about is transcription. So that's basically turning DNA into an RNA. And then we're going to talk about translation, which is the RNA template being turned into a functional complex protein. So first, we're going to talk about that first process, how euchromatin is turned into heterochromatin. The first step is nucleosome formation. So again, we talked about nucleosomes. It's made of a 200 base pair DNA strand. So this 200 base pair DNA strand, DNA is going to be negatively charged. So there's going to be 200 base pairs, right? 146 of these base pairs are wrapped around histone cores, and these histone cores are positively charged. And we know that negative charges attract positive charges, vice versa. So it's really, really strong. It's pretty stable. So this linkage between the histones, the histone cores, and that DNA strand is pretty strong, with 146 base pairs wrapped around those histones. And then the remaining 54 base pairs are used as a linker strand to connect to basically the next nucleosome coming along. So the whole entire strand is a 10 nanometer euchromatin, and this compacts DNA into 1/7 of its original length. So that's very, very like good on storage. So this is the original packaging of DNA chromatin. So again, euchromatin is the original packaging, the loose form, and then it looks like beads on a string. Okay. Second step, you're going to form a solenoid. So first thing that happens, the H1 linker histone will bind to the linker strand, and then part of that core strand, and then it'll start packaging the nucleosomes even closer together. And this will form a 30 nanometer 6 nucleosome coiled solenoid. What this means is there's 6 nucleosomes coiled together. It forms a solenoid, and this solenoid is 30 nanometers. And this all results from the nucleosomes turning into a solenoid. This compacts the original DNA consisted in all 6 nucleosomes in that solenoid 40 times. So it's a 40 times compaction. Now, we talked about a solenoid. A lot of other people believe in the alternate formation. So there's a zigzag formation. So you could either have a solenoid formation or an alternate formation where it looks like a zigzag model. So in the solenoid model, the linker DNA, we're going to assume that it bends. However, in the alternate model, we're going to assume that the linker DNA can't bend because the DNA backbone is pretty inflexible. So it's not going to bend, it's going to stay straight. So in this model, the linker is going to connect the linker histone, I believe, and the linker DNA is going to connect opposite nucleosomes, whereas the solenoid model will connect adjacent nucleosomes. So research shows that both models can actually simultaneously exist. So we're not going to disregard the solenoid or the alternate formation, but we're going to just assume that both of these are equally relevant. Okay, so the third step in that transition is higher order coiling. And there's two types. So that fourth or the second type will be that fourth step. So now we're going to talk about the third step. So it's going to higher order coil into a 300 nanometer fiber chromatin loop. So this is an intermediate step of that higher order coiling where that 30 nanometer solenoid or zigzag, it doesn't matter, it'll coil into a more like it'll be bigger. So it'll be 300 nanometer loops. So it'll be like this and it'll look like a wave on top of a base, basically. So they're going to be 300 nanometer loops with transcription accessible regions, and this is anchored to a scaffold, and the scaffold is made of topoisomerase 2 and 15 proteins that aren't histones. So it's basically like a scaffold where like all of these loops are being anchored to. Now, the last step of this transition from euchromatin to heterochromatin is higher order coiling into a 700 nanometer fiber metaphase chromosome. So this is going to be the official chromosome. This is the final condension step of turning euchromatin into heterochromatin. Turns into a 700 nanometer fiber chromosome, and it is finished in metaphase of that cell cycle. Speaking of the cell cycle, we're going to talk about each part of that cell cycle and how like that chromatin forms. So during interphase at G1, the fiber is going to be 2 to 10 nanometers. So this is when DNA will turn into that nucleosome, like we talked about. During interphase in S, this DNA is replicated. During interphase in G2, it'll be a 330 to 300 nanometer fiber. So this is basically when that nucleosome turns into the solenoid and then also will go into those 300 nanometer loops. Now, prophase is when the 700 nanometer heterochromatin it will be formed. It's not finished, obviously, but it's condensing. It's starting to condense, and then the spindle is about to like assemble. And then metaphase is when the 700 nanometer heterochromatin chromosomes will be finished. So you have 2 times 700 nanometers, so that will be 1,400 nanometers of DNA for that chromosome because remember you have two chromatids joined together, that'll be 2. So this is when the spindle arranges the chromosomes at the equator. And then anaphase is when the sister chromatids separate. So again, 1,400 divided by 2 nanometers. That means there's 700 nanometers each per chromatid. And then telophase is when that cell plate forms and then it'll split. So pretty easy. Karyotype is a key of like all the stained metaphase chromosomes, and it's based off of like chromosome pictures during metaphase, and these are assorted by size, number, banding pattern, as well as centromere position. And just a quick note, heterochromatin stains darker than euchromatin, obviously because it's more condensed, so it's going to look more bright or dark. You have classic or GEIMSA staining, and that's black and white. And then spectral or fluorescent staining is color staining. So I don't know if you need to know that, but fun fact. Okay, so next we're going to move on to what defines the complexity of organisms. The answer to that is the number of cells, the type of cells, and cellular organization. However, it is not that simple. You have the C-value paradox, which basically says that like the amount of DNA that you have, like excess DNA in the genome, it doesn't really mean organismal complexity. So a nice little example of this is that onions actually have more chromosomes than humans do. They have like a really high exponential amount of chromosomes than we do. However, obviously, onions aren't more complex than humans. So keeps you wondering why like okay, so what actually causes the complexity of organisms? Okay, secondly, you have that G-value paradox. So that first C-value paradox was more about the amount of DNA you have, like the genome size. Now, the G-value paradox is, well, sorry, paradox is the number of genes that you have per like DNA. And this basically is like gene number does not mean organismal complexity. So example of this is watercress and also nematodes. They have the same number of genes as humans do, and obviously we're still more complex. So basically, number of cells, type of cells, and actually the organization of cells is what defines the complexity of organisms. Okay, so you have types of DNA nucleotide sequences in the genome. You have three types. You have highly receptive, which is HR, moderately receptive, which is MR, and then single copy DNA sequences, which are unique. So it'll consist of 10% HR, 30% HR, 10% unique, which means like there's 45% nucleotide sequences in the genome. The other stuff is either junk or like probably introns or something like that. Okay, so first we'll talk about highly receptive. So these are at variable lengths at heterochromatic regions around the centromere or the telomere, and these are present at 10 to the power of 6 copies of DNA. And it's basically serves as junk. Like there's literally like they don't do much. Example of this would be alpha satellite DNA with 171 base pair tandem repeats. Um, like it's really it doesn't really do much, really. Okay, so next you have that moderately receptive DNA sequence, and that's 300 base pairs, and it's in euchromatin present at 10 copies of DNA. This includes redundant genes, RNA, and proteins. Example of this would be microsatellite DNA and interspersed repetitive DNA that can move around in the nucleus. So microsatellites are variable amounts of tandem repeats. They're basically all you need to know is they're tandem repeats, okay? And they're tandem repeats in non-coding regions, which are probably like introns or something. And these are polymorphic and useful genetic markers. So it's good to know wherever those repeats are. So a di like microsatellite would be like CA. It's like that type of tandem repeat. Trimicrosatellite is like TCA, and then tetra microsatellite again TCAG, and TCAG. It'll just look like the prefix. Now the single copy DNA sequence, which is unique, this is in chromatin and is present at low copies or even no copies. And this an example of this would be genes or genetic coding regions. So introns would probably either be highly receptive, and then like markers are moderately receptive, and then genes are single copy DNA unique. Okay, so what is a gene? A gene is basically the genotype, like the nucleotide sequence that carries genetic information to express, so which is a phenotype. Every person has one mother gene copy and one father gene copy, so two in total. And there's only like a zero point 0.1 genetic difference in all people. So everybody's pretty much all similar, except for that 0.1% that will contribute to unique physical features. So gene structure, um, this is the structure of a gene. I drew it all the way up here. So it's kind of long, okay? So you read it from 5' to 3', and it includes the distal. I actually don't know what that says. Okay, sorry. Distal controlling factors and then proximal controlling factors. Okay, so first we're going to talk about. Hold up. We're going to talk about the distal controlling factors. Don't even worry about those. Okay, so like they're just like more like enhancers, or like basically reg sequences. Okay, so the first thing you'll encounter when you come from 5', you'll see that GC, and that's basically the GC box. And we'll talk about that in just a bit. And then right after the GC box, you'll see the CAT box, which will be CAAT. These two are regulatory sequences, and they're basically enhancers. So the CAT box is, or sorry, let's first talk about the GC box. So the GC box is bound with activator proteins to activate gene transcription, so that's really important. And then the CAT box is responsible for inducing gene production, so that's also really important. So the GC box and the CAT box are proximal controlling factors, which just means that like they're closer to the actual gene. Well, distal controlling factors are on the outside. After those two, you'll find the TATA box. So basically, the Tata box is the promoter, and it helps begin the signal of basal transcription. You can find this right before the start codon. So the GC box, cat box, Tata box, all of these three consist, or they make up the upstream regulatory sequence. What are regulatory sequences? They're site of regulatory proteins that affect binding and transcription rate. So all the proteins bind there. They're not going to bind on that gene. They're going to bind on where the sequences, the regulatory sequences are. And it also helps with transcription. So after that Tata box, you see that start codon. After that start codon, it'll be the start of the gene, like the whole gene in general. So the gene will consist of basically a reading frame. So an open reading frame, okay, basically a reading frame is one of three ways to read a gene. And then an open reading frame is a sequence between the start and stop codons. And this whole open reading frame is used for transcription and translation. And an open reading frame consists of exons and introns. Introns are intervening sequences. They basically are useless. They're just there to help stabilize the gene. Now, the exon is really important because it contains the gene. The first exon will contain a UTR, which is a 5' untranslated region that, oh, sorry, the 5' untranslated region is right before the start codon. So, yeah, sorry. So after that start codon, the actual important part of the exon will begin, and then it'll be like exon intron, and then at the very end, you'll see another untranslated region, and that'll be the 3' UTR. It'll be after that stop codon. So after the stop codon, you'll see the 3' UTR. After the 3' UTR, you will see the poly A signal, and that poly A signal will basically be followed by the terminator. So the terminator will signal the end of transcription. Again, exons and introns are the transcribe region, but the transcription is going to end after the poly A signal after that termination. So that'll like after that terminator, and then you'll have distal controlling factors. The terminator and the distal controlling factors on the 3' end are part of the downstream regulatory sequence. So that's basically the whole anatomy of a gene. Okay, so we have types of genes. We're going to talk about some of those. The solitary genes, these are basically a unique single haploid copy of a gene and consists of like the bulk of euchromatin. Duplicated genes are a portion of the chromosome that is copied, or a paralog. Either copy might mutate due to meiotic errors. And then multi-gene families are sets of similar genes formed by duplication in similar chromosome sections. Pseudogenes are dysfunctional gene relatives because of multiple mutations. And then repeated genes are like high amounts of copies of small genes that are clustered at specific sites in like mRNA and tRNA, and they have a head to tail configuration. Okay, so now we're going to move on to the central dogma of molecular biology, which is basically DNA genotype to RNA intermediate to a protein phenotype. Very, very simple way of saying it. However, it's a lot more. So how you explain this in more detail is basically transcription and translation. So we're going to start with transcription. This is when DNA will turn into RNA. Well, DNA doesn't turn into RNA. DNA is a template served to create an RNA strand, basically. So this is performed in the nucleus during G1 or S or G2 or M. So there's transcription wave. So it can start at G1 and at S, or start at G2 and at M, you know? Okay, couple of steps that comes along in this process. The first step is pre-recognition. So this is when euchromatin, it has to be like converted. So like heterochromatin must be turned into euchromatin before transcription. So how heterochromatin, this thick chromatin, will be turned into euchromatin, it is turned through lyase K acetylation. I can't really tell you how it works, but all I know is that like basically this histone acetyltransferase, which is like HAT, HAT, histone acetyltransferase, and then histone deacetylase, which is HDAC. So HAT and HDAC, this will both decrease the positive charge of the histone. So if you remember, like the DNA strand is usually negatively charged. Now the histone is positively charged, but if HAT and HDAC decrease that positive charge of the histone, the DNA is not going to really be attracted to that. It's basically like if you like conduct like ugly facial surgery and your boyfriend becomes less like interested in you. You know, you see like if you're pretty, right? And then your boyfriend has a good like a big attraction to like pretty girls, if you make a ugly like surgery, he's not going to be more attractive. So that's basically that's what happens to the charge of the histone. It becomes less positive and it becomes more negative. DNA is not going to be attracted to a lessly like positive histone. It's going to want to look for more positive. So that means the DNA strand is going to be loosened from the histone, making heterochromatin, which is tightly condensed, a lot more loose. That's basically what happens at pre-recognition. And then hold up, let me check and see if it's true. Okay, yeah. I'm not being stupid. Okay, the next part is recognition. So the 100 protein complex, or the PIC, this is formed, and the PIC needs to be formed for RNA polymerase 2 to bind. I wrote it in here as PNA. Give me a second. Okay, so you know when we talked about the Tata box, what we need, that's basically a promoter right before the untranslated region and the start codon. So that Tata binding, that Tata box is really important. It's a promoter region, and the Tata binding protein, which is the TBP, will bind to that box and then recruit the transcription factors, which is like TFIID. So that's the second part. Third part is initiation. So this is when the 20 protein mediator complex is formed and then RNA polymerase 2 binds. So the first step is RNA polymerase 2 and other transcription factors, including TFIIB, TFIF, and TFIH. These will bind to that, like to that part. And then ATPase and helicase will unwind DNA into a transcription bubble. So you know we like talked about that transcription bubble when it like opens up. That's basically what that is. And then RNA polymerase 2 will bind to that template strand active site. And this is basically like an initiation of transcription. Okay, next step is elongation. So RNA polymerase 2 will move down the template strand to read it 3' to 5', and then it'll transcribe 5' to 3'. So how this starts is RNA polymerase 2 is unphosphorylized or unphosphorylated at the carboxyl terminal domain, the CTD, and this is basically done by the mediator complex. The mediator complex will unphosphorylize the polymerase 2 at the CTD. That will start the elongation, and then it'll start reading 3 to 5 and then transcribe 5 to 3. And then basically the copy strand replaces thymine with uracil, and then it'll replace the DNA's deoxyribose with just like simply ribose. Okay, lastly, you'll have termination. So this is a cleavage of the new transcript that's made from elongation. So CPSF is in charge of polyadenylation and cleavage, and then CSTF is important for cleavage. So both of these transcription factors are carried by CTD, and they both recognize the poly A signal. Like I said, at the very end of this sequence, you have that poly A signal, and then give me a second. Let me search up, is the poly A signal after the stop codon. Let me do that because I don't want to lie, and it'll be embarrassing if I get that wrong. Okay, so while we talk about that, the other, it'll bind to the poly A signal, and then that'll indicate the cleave area. So other proteins are recruited to carry out the cleavage, and then the strand is cut downstream of the poly A signal, and this will form hnRNA. Okay, and also the poly A signal is also right after that stop codon. So just to let you know, that's how it is. Okay. Now, after termination, that's not the only thing that happens. You have an afterlife, okay? So it'll start adding stuff. So like, you know how a car is made? Like it's everything is done, production has terminated, but the new owner, they're gonna want to do car mods, you know, to make it cooler. So this is basically car mods, but it's RNA mods. So basically there's like three types. So you have 5' capping, and it's like it's literally a cap right in front of everything. So it's like it's gonna be like a guanine cap. So this happens co-transcriptionally. When the RNA is about 30 nucleotides long, a guanine group is added to the 5' end by capping enzymes. So the function of this is for nuclear transport, and this actually promotes the first step of translation. We'll talk about that in a little bit. And it'll prevent degradation of that RNA, and then it'll play a part in intron splicing as well. So, what is splicing, you ask? You didn't ask, but I'll tell you anyways. So, this is basically RNA processing that happens both co-transcriptionally and post-transcriptionally. Oh my gosh. And this is when introns are removed from the sequence and then exons are ligated, and this will result in mRNA. So, basically, hnRNA is immature because it contains both exons and introns. So it'll go through three processing steps to become mRNA. So SNRPs or snRNPs, this includes U1, U2, U3, U4, U5, etc. These will bind to specific splice sites at intron-exon boundaries, and this will aid with precise splicing and ligation. We'll talk about this whole splicing processes here in just a little bit. And then it'll result in mRNA, which is mature RNA. It's also known as messenger RNA, but just think of it as mature RNA because it's better. Because the mRNA will have the 5' cap, 3' prime tail, and then exons only, no introns. And it'll be ready for export to the cytoplasm. Okay, so what is that 3' tail? So this is made through 3' polyadenylation. This happens right after transcription, like right after the cleavage happens. So up to 200 adenines, and will be added to the 3' end. So this is gonna end up in a polyadenine tail, so a poly A tail. And this is done by poly A polymerase. And the function of this would be mRNA stability and transport to the cytoplasm. So that's also really, really important. Some little transcription rules. Each DNA strand is separated to make that bubble, but only one strand is the template. Each gene has its original transcription protocol, and nucleotides can be only added to the 3' end, and it will result in an anti-parallel and unidirectional RNA complementary strand. Okay, now we're going to talk a little bit about introns because they're kind of important. Like they're kind of whack, but like they're removed, but they're not really important, but the way that they're removed is important. So that's what we're talking about. Okay, so 4 types. So they're either in protein coding genes or hnRNP, and these are removed by spliceosomes, or they can be in tRNA genes removed by proteins, or they can be part of that group 1 self-splicing. They don't need a spliceosome or proteins, but they do need a guanosine 5-triphosphate, which is GTP, to catalyze their own removal from the mRNA or uTRNA or rRNA precursors. Group 2 self-splicing introns are like they don't need GTP, but they do need a little bit of protein assistance to self-splice. Okay, now we're going to talk about like anatomy, I guess, of an intron. So from on that 5' end, you'll meet an exon, right? But we're not talking about exons. But I'm just saying like introns are in the middle of exons. So there's 2 exons, and in the middle of the 2 exons is an intron. So first thing you encounter at an intron is the GU site, and this is basically the 5' splice site. It'll identify that first splice. And then you'll a little bit more downstream, you'll meet the branch point sequence, and what the BPS looks like, it's UAC, UAAC. So UAC, UAC. This is the binding site for SNRP2, SNRP U2, I guess. And then downstream from that, you'll see the polypyrimidine tract, also known as PPT. It'll be a whole bunch of pyrimidines, and this is going to promote spliceosome assembly. This is right before the 3' splice site. Now, right after that, at the 3' splice site, you'll see the AG nucleotide acceptor site, and then that will be the end of that intron. Okay, so now we're going to talk about how a intron is spliced. So first, SNRPU1 binds to that 5' splice, and then SNRPU2 will bind to the BPS, and then SNRPU4, 5, and 6, that's the trimer, will be recruited to that 5' splice as well. Now, after that, U1 and U4 will dissociate from hnRNA, and then U2 and U6 will step in and replace that. A lariat, or like basically a lasso shape, is going to be formed. And then right after that, the 5' splice is cleaved by that either U1, or sorry, is cleaved by U2 or U6. And then U5 will bind to that 3' splice and cleave that. And then U5 stays there and ligate the exons together. And then after that, all the SNRPs are going to be released with that intron, and then that intron is like lasso shaped. It's circular, you know? Okay, so that's regular splicing. Now let's talk about a little bit about alternative splicing. This is a regulated process when like a bunch of different exons are joined. So you know how like the exon ligation is usually like right after, like, if exon 1, 2, and 3 are cleaved, they're going to be put together as 1, 2, and 3. Alternative splicing is when there's a different organization of exons. So like a single gene can have multiple mRNA variants because there's different protein isoforms because of a different combination of exons. So instead of 1, 2, and 3, you'll have 1, 3, and 2, 2, 1, 3, 3, 1, 2, etc. This is also known as exon skipping. This also increases genetic diversity and is the same mechanism as spliceosomes under specific factor controls. So cis factors are inside of the gene and regulate gene expression. So this would be promoters and enhancers, etc. Trans factors are diffuse proteins that regulate gene expression. For example, SNRPs, and transcription factors and other. And then the effects of this can be either primary splicing defects. This results from cis factors, and it'll end up within a defect protein product. Secondary splicing defects result from trans factors, and these end up with mutations due to splicing issues. Okay, before we talk about translation, let me talk a little bit about tRNA. So tRNA has anticodons that match with mRNA codons. Like basically, codons are like 3 sets of nucleotides that will code, like each codon will code for one amino acid. So, for example, AUG will code for methionine, and this will initiate like a start codon. Now, UAA, UGA, and UAG, let me see what they code for. I know they code for a stop codon, but let me see what amino acid they code for. Maybe they don't even code for amino acid. They're termination codons, and I guess they don't. Right, they don't have an amino acid. I'm being stupid. Yeah, because they don't have like a tRNA for them. Okay, whatever. We're gonna be smart now. Okay, so now we're gonna move on to translation. This is when mRNA will be converted into a protein in the cytoplasmic ribosomes during G1 or G2, depending on when transcription ends, basically. Okay, so activation is the first step. This is when amino acid is charged to tRNA. So basically, tRNA is linked to their corresponding amino acid. So for example, if mRNA says AUG, that tRNA will have that anticodon, and that anticodon will correspond with a specific amino acid, in this case methionine, and this will be conducted by amino acyl tRNA synthetase. Sorry. This process requires ATP, and the result will be an amino acyl tRNA. Why is it called that? Because it's tRNA linked to an amino acid. So the amino acids are covalently linked to that 3' end of the tRNA. This is basically charging. And then recognition features will ensure that the correct amino acid is added to the tRNA. The third base variable position will allow some tRNAs to bind to even more than one codon, which is kind of nice. And then that next step will be initiation. So this is when ribosomes will start binding. So the 40S ribosome will bind to that 5' cap at mRNA. Give me a second. I feel like we're missing something. Give me a second. We'll talk about it later. Okay, well, actually, we'll talk about it first. So let me talk about how like a ribosome looks like how the ADS ribosome looks like. So this is the ADS ribosome. It looks like a jellyfish, I guess, like a jellyfish head, and then there's like 3 buttons on it. Okay, so that bottom base is that bottom base will be the 40S small subunit, and this is in charge of reading the mRNA strand. And then on top of that will be the 60S large subunit of the ADS ribosome. And this will form amino acid chains by putting peptide bonds in between them. And then the 3 buttons, you first have the E site, the P site, and then the A site. The A site is where that amino acid tRNA will enter the ribosome and be like put into place. And then the P site is where it'll move on, like it'll move from the A to the P. And then from that P, it'll be like peptide bonded to that to another new amino acid tRNA in the A site, if that makes sense, right? And then when it's done, when it's cleaved, it'll be cleaved from that P site. And then you're wondering like, okay, so what I mean, like that first site is the E site. So the E site is where the tRNA, like since like after the peptide bond is like formed, you like tRNA is kind of useless, like the bond is formed, it's going to dissociate from that strand, and then it's going to move on to that E site and then be released into like the atmosphere to repeat the process basically. So that's basically what it looks like. So during initiation, that 40S ribosome subunit will bind to that 5' cap at mRNA. Remember we talked about that guanine cap? It's going to bind to that cap at the mRNA, and it'll scan for that start codon at mRNA. So yeah, so 40S ribosome will start reading. The amino acid tRNA synthetase, oh my goodness, this will attach methionine to tRNA, and this will create met-tRNA. That's the methionine is the amino acid for the start codon, and this will bind to the start codon at the P site of ribosome. Again, the P site is like the middle site. And then the binding of the start codon will actually recruit the 60S large subunit, and this will form the ADS ribosomal complex, which is like the jellyfish. With the ADS ribosome assembled, then the met-tRNA will occupy the P site, ready to form a peptide bond with the first amino acid tRNA entering the A site. Basically what I just explained. Okay, next up, elongation. Oh my gosh, I was about to say Elon Musk. Okay, elongation factors. You have 2 of them. So both elongation factors, which is EEF and GTP, will both allow entry of the charged tRNA into the A site, which is the entrance site. So EFTU or EF1A in eukaryotes, this consists of 6 proteins and will catalyze addition of the correct amino acid tRNA and the correct codon and anticodon match to the A site. And it does this, it only catalyzes it, doesn't do it. It catalyzes the addition of the AAtRNA to that A site. Well, it catalyzes this by hydrolyzing GTP, so it'll turn GTP to GDP, and this will provide energy for that whole process. EFTS or EFIB or 1B will act as a guanine exchange factor or GEF. So when the EFTU has hydrolyzed the GTP to GDP, the EFTS will take away that GDP and replace it with a new GTP on EFTU to charge it basically for continuing the cycle, So it's like if I drank all my water and I have an empty water bottle and somebody gives me a new water bottle in exchange for that empty water bottle. That's kind of like not really practical, but I'm just saying this is basically what's happening. Okay, so second step of elongation, the initiation factors will ensure the correct charged tRNA enters the A site. And then when it's at the A site, the GTP hydrolysis will release the phosphate and provide energy to release elongation factors. The peptide bonds will form between the amino acid in the P and the A site. And then the amino acid originally in the P site is released from the tRNA being attached to it, and then it'll extend the polypeptide chain by one amino acid. So the ribosome will move and translocate down the mRNA strand by one codon, shifting the tRNA with the peptide chain from the entrance at the P site to the A site to the P site, and then after that the tRNA is released. So that's basically what I just said. I don't know why I said that twice. It's weird. Okay. Anyways, after that you have termination. So this is when the stop codons, or UAA, UGA, or UAG, will signal termination. Now there are no tRNAs for stop codons, which means there are no amino acids for stop codons. They're just simply stop codons. After you see that stop codon, you will have release factors that bind to that A site and release the chain from the A site. This will dissociate the ribosome from the mRNA. Okay, we're talking about translation rules just a little bit. Again, the 60S large subunit will consist of 3 rRNAs and then 46 proteins. These form peptide bonds. 40S small subunit will consist of 1 rRNA and then 33 proteins. This is responsible for reading mRNA. The E exit site is where the tRNA without the amino acid moves to be released from the ribosome and recycled. Then the P peptidyl site will hold the tRNA that carries the growing polypeptide chain. And then the A amino acyl site is where the charged tRNA with the amino acid moves in to the ribosome to pair with the mRNA codon being translated. So basically, okay, listen, mRNA does not touch this ribosome. Okay? The strand also does not touch this ribosome. The only thing that touches the ribosome in the areas, in the sites, is the tRNA, right? aRNA, that's where the tRNA with the amino acid is. And then P site is where that tRNA moves and it carries that growing chain. And then the E site will move to like that's where the tRNA moves and it'll dissociate from that strand. So basically all of this happens from 3 to 5, but not really, because it's like moving this whole ribosome thing is moving from 5 to 3, but it looks like the strand is moving on the ribosome from 3 to 5. Like, if that makes sense. But if you're looking at a video from the strands like viewpoint, it's moving from 5 to 3, but for the ribosome, if you're looking at that, and it's moving from 3 to 5. Okay, it's looking kind of confusing, so I'm gonna stop talking. Okay, so test question: a peptide has 100 amino acids. How many codons does it have? Okay, well, since it has 100 amino acid, it'll have 100 codons. However, you have to account for that stop codon because, sorry, you have to account for that stop codon because there's no amino acid for that stop codon. So just because it has 100 amino acids doesn't mean it has 100. It does have one more codon, and that will be 101 codons in general because that stop codon doesn't have an amino acid. Okay, the number of anticodons will be 100 because it'll be 100 codons and then 100 anticodons, and one of those 101 codons won't have an anticodon because it's a stop codon. The amount of tRNAs needed for translation, again, will be 100 because the stop codon doesn't have a tRNA. And then the number of nucleotides needed for translation will be 303 because you have, again, 101 codons, and then you have 3 nucleotides per codon. So that would make 303 nucleotides. All of this, and we still haven't talked about all of this stuff. Okay, anyways. Okay, almost done. Proteins are macromolecule gene end products, and the functions can either be binding. For example, TBP will bind to the promoter region or the Tata box, and it'll be a platform for transcription initiation. Proteins can also be part of catalysis. The example would be DNA polymerase. It catalyzes DNA replication. Another function will be switching. For example, GTPase proteins for example, RAS, will help signal transmission because it'll bind to GTP to signal the active state and switch it off when GTP is hydrolyzed to GDP. So when it is hydrolyzed to GDP, it is an off switch. Now, if it is GTP, it'll be an on switch. Proteins can also act on structure. For example, silk is made of F-actin. Okay, we're gonna talk about protein versatility and then protein structure, and then we'll be done. No, we'll be talking about protein stability too. Oh, I'm sorry. Okay, protein versatility will come from chemical diversity, chain flexibility, and interaction diversity. So first, you have that chemical diversity. So there are 20 unique amino acid side chains and interactions. So first you got those nonpolar or hydrophobic reactions. This will repel water and pack closer to each other. So you have ALA A, Val V, Leo L, Illa I. Okay, sorry, it'd be funny if I say it like that, but that's the abbreviation. So alanine, valine, leucine. Okay. Secondly, you have polar or hydrophilic not reactions. Bonds, interactions. So these will be like hydrogen bonds with each other, or or bonding with the peptide backbone. So this will be glycine G, serine S, theine, I guess, THET, and then cysteine C, and then you have charged. So these will be outside of the protein and interact with other molecules to create a 3D structure. So positive amino acids include LYSK, arginine R, histine H. Negative amino acids include ASPD and then GLUE. It's funny because it's like it spells kind of like glue, GLUE, but it's actually GLU-E. Okay, anyways, the second part is chain flexibility. So there's a peptide bond, and that's basically a covalent bond between carbon and nitrogen. This will create a coplanar peptide plane. So it's like a strict bond, I guess. But it'll allow free rotation of nitrogen to alpha C or C to alpha C single bonds. So these single bonds, of course, are going to be free to rotation. So that's why proteins can have a really cool structure or function, because structure equals function. Nextly, you have interaction diversity. So you either have covalent bonds or electrostatic bonds or interactions. So covalent bonds are strong and stable interactions from electron pair sharing. So examples of these would be amide bonds of the amino acid backbone. So alpha C to C again. Or you have disulfide bonds between cysteine. So cysteine and then sulfide, cysteine. And this will be for stability. Okay, and then nextly you have electrostatic bonds, which are weak interactions. So like van der Waals, Okay. Next, you have protein structure. Primary will be linear amino acid sequences. These are connected by covalent bonds. There's two ends referred to the amino or the nitrogen terminus, and then the carboxyl or C terminus. Okay, so secondary structure will be protein substructures, and these are based on hydrogen bonding. You have beta turns, which are 3 to 4 amino acids, and they are compact. They help in compact folding. Beta sheets are pleated strands in primary sequence, and they're oriented side by side. And then the alpha helix is most common. And then you have tertiary, which is folding of secondary elements into a more compact and nearly solid object, stabilized by electrostatic interactions, which are polar, nonpolar, and charged interactions. Okay, quaternary structures are 3D multi-unit complexes. Okay, next. Protein stability of tertiary structures are determined by covalent bonds, electrostatic interactions, post-translational modifications, protein motifs, and protein domains. So first, or third, I guess, post-transitional modifications include that disulfide bridge, again, like covalent bonds between cysteine and sulfur. And then metal binding is basically metal with a amino acid side chain. And then you have binding of effector molecules, for example, glycosylation, which is the addition of carbohydrates to SARE, THR or ASN to help stability. Then you have phosphorylation and acetylation, which will help activate or deactivate stuff and helps with binding affinity. Then you have ubiquitination and methylation, and this will help conformation and protein function. Then you have protein motifs, which are they basically help binding, you know. So primary sequence motifs are found in DNA binding proteins. Contiguous secondary functional or structural motifs, it can be like a Rossmann fold, and this also helps in binding, like with like protein or like small structure binding. Okay, and then you have protein domains, which are specific protein regions within a protein with stable folding and is independent folding from the rest of the protein. So there's this independent structure and function from the rest of the protein. So example would be an SH2 domain. Okay, so first we talked about that central dogma, which is basically DNA to RNA to protein. After this whole thing, we learned that the central dogma basically is more like nucleotide sequences. So, like DNA will be used as a template to be turned into RNA. And then from RNA, it'll be used as a template to create an amino acid sequence polypeptide chain. From that, you'll have that amino acid sequence polypeptide chain with the interactions, and this will form a fully structured protein. And then this will basically form a fully functional protein in the body. So that's the altered dogma, I guess. Hopefully it makes sense. Bye, I guess. Oh my god.